On the protein-centric pages, we have a section dedicated for ligand binding sites and ligands that are observed to be interacting with the protein of interest. This section is made of two main parts. At the top, we have a gallery, and underneath that, again, we have a ProtVista sequence feature viewer. At the top, you can see two numbers. One is the number of directly interacting ligands, unique ligands or distinct ligands, and the other is all the available ligands. The difference is that on one hand, you have all those ligands which are directly interacting with the protein of interest, meaning that in the PDB entry, you see that this chain is interacting directly with these molecules. On the other hand, you see all the ligands which are observed in the same PDB entry as the protein of interest. There is a search field which you can use to filter the gallery, this being the gallery, and you can use either the molecule name, the unique molecule code, which is we refer to as head code, or the PDB identifier. Using any of these, we filter the gallery underneath. So for example, starting to type in sulfate, it will filter down the gallery to that specific ligand. By default, we are showing only those ligands which are directly interacting with the protein of interest, but you can tick this box and then you will be able to see all of them. So by default, we only show these 18 directly interacting ligands. Clicking this, we show you all the 81 ligands. If you mouse over or tap on any of these unique identifiers for the ligands, then you will be able to see the full name of that ligand. Under that, you can see a link, and in fact, this link goes to the PDB search, and it will list all the relevant PDB entries where you can see this ligand together with the protein of interest. And finally, if you click on the small image of the ligand, then you will get to the 3D visualization. This will be by default focused on the first instance of this ligand occurring in contact with the protein of interest. It can happen that you have this ligand in more than one PDB entry, and in this case you will see how many entries is this ligand observed in. And in that case, if you go to the 3D visualization, you can use this drop-down to choose which PDB entry you would like to see. Under the gallery section, we have the sequence feature viewer. And here we are showing all those residues of the protein sequence which are directly interacting with a number of ligands. If you click on any of these highlighted segments, then you will see more information regarding which residues are there, the range of them, also which ligand are they binding to, and an example entry where you can go and see the details. If you collapse this track for the ligand binding sites, then you get an overview of where all these different binding sites are located, and you can discover the pattern of those residues which are consistently binding to small molecules or ligands. And finally, if you would like to download this data that's being displayed in the sequence feature viewer, then you can choose between JSON and CSV formats.